Okay, welcome back, ladies and gentlemen, from an abbreviated lunch. Thank you for cutting it short, and we'll probably have some additional attendees join us in the next few minutes. We are doing a panel on NASH to MASH. It's a unique panel in the sense it's the only specific disease-oriented panel of this prix Galien. And I want to thank the organizers and Mike Rosenblatt specifically for inviting me to chair this session. I'm Dr. Alan Moses. I'm an MD, an endocrinologist, not a hepatologist. I had an academic career at Harvard Medical School in diabetes, was chief medical officer of the Joslin Diabetes Center, and then joined NovoNordisk in 2004, where I stayed for 14 years. Leaving as the global chief medical officer in 2018. Since then, I've been a consultant to the biopharmaceutical and pharmaceutical industry. The topic we have today is actually a really interesting one, and we've got a very impressive committee joining me because they cover all aspects of this disease, from the patient orientation of a patient advocate or a patient advocate organization to academic physicians who dive deeply into mechanisms of action of both drugs and hormones within the liver that may be targets for the disease we're going to be talking about, and then 3 individuals from the pharmaceutical industry who are deep into clinical development now, both discovery and now clinical development of molecules that are targeting MASH as a therapeutic indication. Before I introduce my panel, and there's so many of us, I can't even see Søren down at the end, especially with the light in my eyes. But before we do that, and because this is disease-oriented, I want to present a couple of slides to give you, who are not familiar with this disease state, sort of a common background so we can understand what's going on. So we're really talking about the evolution of disease from both a pathophysiologic perspective and a public health perspective, and possible solutions. Again, both from an intervention, that means pharmaceutical intervention, but also perhaps public health measures as well. For those of you who are old enough to remember the 1950s, this is a NASH. But like the automobile, the name of non-alcoholic steatohepatitis is no longer going to be used. Rather, we've moved on to something else. Metabolic dysfunction associated steatohepatitis. Now, this name change has occurred over the last couple of years. In fact, so recently that the first drug approved for the indication bears an indication for NASH, not for MASH. But it's important. And I'm not even sure this is the best name. I would actually call it DASH, or dysmetabolic associated steatohepatitis, but we won't get into that argument. The implications for MASH are serious. It's a serious disease with high prevalence, and without a slide, I can't see. There we go, thank you. Limited therapies, and we're going to talk about that today, obviously. It's got association with multiple other serious conditions, and we'll talk about that. And importantly, very importantly, low appreciation among the public and clinicians. Indeed, it's projected to be the #1 cause of end-stage liver disease requiring liver transplantation over the next 5 to 10 years. Its prevalence is driven by obesity and diabetes obesity, but there clearly are individuals who have this condition who have neither. And we'll talk about that a little bit as well. Just to give you a sense of what the disease looks like, it is a liver disease by definition. On the left here, you see what a normal liver looks like with fat accumulation, excess adiposity within the liver, fat accumulation. You have a condition called MASLD, which is metabolic associated liver disease. Then you move to MASH, where you begin to see fibrosis and bridging and other pathologic findings that you can see under the microscope and using some other technologies. And then that degree of fibrosis moves on, fibrosis and inflammation that ultimately moves to the point of cirrhosis and end-stage liver disease. There are a number of predisposing factors and there are a number of pathogenetic factors that really complicate our full understanding of the molecular pathogenesis of this disease. But if we think about it from a public health perspective, it is unfortunately all too common. In individuals who are not overweight, about 30% have evidence of a little bit of excess fat in the liver. The clinical implications of that are unclear. About 20%, about 18% of the general population have some evidence of MASH, that is, fat and evidence of fibrosis. More severe fibrosis occurs, fortunately, much less frequently in the general population. However, in the obese population, and you could substitute obesity/diabetes here, 60% have some evidence of excess fat in the liver. Okay? 35% have evidence of MASH, significant disease, and a significant percentage have evidence of MASH with fibrosis, disease that should be intervened with. So understanding the pathogenesis is critical to diagnosis and treatment. The primary goal, of course, is prevention. That's what we're not necessarily talking about with pharmaceutical intervention, although maybe with a caveat. The secondary goal is to base your treatment on the underlying intrahepatic metabolic dysfunction. Because increasing lipid in the liver induces inflammation, then fibrosis, and then more severe fibrosis, and ultimately cirrhosis. Until recently, there were no approved therapies. There now is one, fortunately. Madrigal was able to get approval for a drug called resmetirom, or Resdifra, in an accelerated approval. And that's changing the landscape of how we think about this disease from a clinical perspective. And there are a large number of molecules addressing different molecular targets that are in development and we'll touch upon today. But there are a lot of unanswered questions as well. What is the diagnostic or regulatory role for liver biopsy in the definitive diagnosis and follow-up of this disease? Should we be focusing more on prevention or intervention at the time the diagnosis is first made? Which patients should be initiated on therapy, or should some undergo watchful waiting depending upon staging of disease? And that depends on how accurately we can stage disease. How long should they be treated? Is monotherapy going to be enough, or do some of these individuals need combination therapy targeting different pathways? From a commercial perspective, what therapies in development are likely to make it to the marketplace? And also importantly, what are the implications of MASH for non-liver organ dysfunction? Because there is good evidence that in fact twice as many people with MASH die of heart disease than die of liver disease. So with that as background, let's begin. But first with an introduction of my co-panelists here. They're not in order, but they're in order on my page. Dr. Ira TABAS is the Richard Stock Professor and Vice Chair of Research, Department of Medicine at Columbia University, Irving Medical Center, and an expert in the underlying mechanisms of MASH and the pathways that can be targeted potentially therapeutically. Dr. Anthony HOLLENBERG is the John Wade Professor of Medicine and President at Boston Medical Center, and his research career has been focused on mechanisms of action of thyroid hormone and how thyroid hormone is involved in metabolic regulation. That's important because of Resdifra. Now we have three representatives from the pharmaceutical industry. To my left is Dr. Stephen Goff, who's the Global Chief Medical Officer at NovoNordisk. He came to NovoNordisk from a role as head of the Oxford Diabetes Endocrinology and Metabolism Center and Professor of Medicine at the University of Oxford. We have Dr. Mark Hartman at the center there. Mark is an MD and Vice President for NASH obesity at Lilly Research Laboratories, Eli Lilly. He received an MD from the University of Connecticut, spent over 10 years on the endocrine faculty at the University of Virginia before joining Eli Lilly. Søren TULLIN is a PhD, Senior Vice President, Global Head of Cardiometabolic Disease Research at Boehringer Ingelheim. Søren received his PhD in biochemistry and molecular biology at the University of Copenhagen and worked for 20 years at NovoNordisk research laboratories in positions of increasing responsibility before joining BI 5 years ago. And finally, but certainly not least, we have Jeff McIntyre, who serves as the Vice President of Liver Health Programs at the Global Liver Institute. Jeff fully understands the challenges of metabolic liver disease and the burden that this disease places on individuals suffering from it. The Global Liver Institute is focused on increasing awareness of liver disease and assuring that individuals, most of whom are asymptomatic, can be diagnosed early enough to make a difference in their life course. So let's dive into things here. One of the major challenges that our system faces in the area of MASDL and MASH is that early diagnosis that is potentially more responsive to therapy and perhaps even reversible is asymptomatic. And clinicians do not routinely utilize available tools to assess the risk of this disease in their patients. So let me turn to Jeff, who as a patient care advocate sees this all the time and spends a lot of his time thinking about it and thinking about strategies. Jeff, who should be screened for MASH as we enter an era of new diagnostics and new therapies that are soon to come to the marketplace?
Thank you, Alan, and thank you to you and the panelists and the pre-Galien for including the patient voice in this conversation. We consider that a priority, and I think we're the only one on the docket for the day today, so we are greatly appreciative and honored to be here. When it comes to who should be screened, there's kind of two answers to this. One is anyone that has a liver should be concerned about their liver health.
That's a little broad, Jeff.
Pretty sure that includes most of the folks in the room as well. In the recent nomenclature change from non-alcoholic to metabolic, I think what we have found is that this serves it helps with some clarity for anybody that has any type of metabolic dysfunction to be screened for liver disease, for fatty liver disease or MASLD and MASH, if you will. We would love to see it as ubiquitous as blood color cuffs, as blood sugar measurements. We believe that it is that prevalent in our society for us to be able to do that. We know that if you have diabetes, you should be screened for MASH. If you're pre-diabetic, you should be screened for fatty liver disease or MASLD. That there are these kind of levels of risk stratification, if you will, that are important to get folks into the system and moving through a care pathway.
And so how do we translate that into education and public awareness for the general public, but more specifically for clinicians who are going to see patients in their office to identify those who should be screened? Because, I mean, you can say that everybody who has diabetes should be screened, but it's not happening. We know that.
It is not happening. I think we've had a lot of finger pointing of primary care needs to step up. We've had a nomenclature change. I mean, to be fair to the nomenclature change, people didn't know NAFLD, they don't know MASLD. And so we need to rise the tide on all of this. If you'll forgive me, let me do a quick test with everybody. Those that are willing, please raise your right hand in the air. And if you would raise your left hand in the air. Okay, now touch your liver. Yeah. Okay, I saw a lot of folks that were like they were back in high school looking at their classmates' answer to see if they got it right, and there was somebody in the back that kind of had a Macarena moment there as well. So we need to do better education around this, and that comes at all levels. Education awareness is always going to be an important part of this, but I would emphasize that it is necessary but not sufficient. Patients and the public are faced with an incredibly complex health system, complex payer and reimbursement system. And the patients need to be empowered through more than just education about liver disease, but also to be able to hold their providers accountable, to hold the FDA accountable, to hold the regulators accountable, and to hold the payers accountable in what goes on as well.
Great.
Alan, can I just say something? Because I'm just looking at the audience, and I think it would be good to have a few little basics here. So there are hundreds of millions of people with fatty liver, and 80% of them will do just fine. So the key issue is to determine who's going to progress. And then that's tied up with how you determine that, because technically speaking, and I'm not a hepatologist, as everybody here knows, but my best friend is a hepatologist. So, but technically speaking, MASH is a pathological diagnosis as we're sitting here today. So we've got to deal with that issue and how we can use non-invasive tests. But the trick is all of those people that have fatty liver, how do you determine who's going to go on to actual liver disease? And that's really a key point, right?
It is. It's a very important point, and I didn't mention during the brief introduction that there are clear stages of fibrosis in MASH also that have different implications. And from a regulatory perspective, this is important in terms of, admitting patients or individuals into clinical trials. We'll get to that a little bit as we talk about the indications for approved drugs and the indications being sought for drugs in development. But there's a lot of controversy about the best diagnostic tests to screen, identify, and track NASDL and MASH. And unfortunately, we don't have time to go into that in great detail. But the gold standard, regulatory gold standard diagnostic procedure has been a liver biopsy. Now, in the panel, first thing this morning, they were talking about women's health. And we apologize, by the way, of being the only, I think, all-male panel up here. So we recognize that, and that's just the way it worked out. This liver biopsy, required by the FDA for drug development at the moment, has been a real impediment in terms of recruiting individuals for trials. Not surprising. Just as in the panel this morning, they were talking about the need for laparoscopic surgery to make a diagnosis of endometriosis. So the real question is, what really is the role of liver biopsy in diagnosing individuals with MASH, and how is it viewed by persons potentially affected by NASH? And again, how much of an impediment is that to the conduct of clinical trials in the regulatory environment? Is there a sufficient level of evidence from other diagnostic modalities that would allow those modalities to be substituted for liver biopsy to enroll subjects and to follow them with sufficiently sensitive and accurate data over the course of treatment? So I'm going to start this with a conversation from a drug development perspective. And so Mark, Steve, Søren, and I don't care which order, how do your companies view and address the need for liver biopsy in drug development for MASH? And do you have any thoughts about how the label for Rezdiffra might open up the opportunities for the FDA to rethink whether liver biopsy is actually required to initiate therapy? Because the Rezdiffra label, despite the need for biopsy to get into the trial, does not require a liver biopsy for initiation of therapy.
So let me jump in here, Alan. So let's help the audience understand why liver biopsies are being done for clinical trials while the FDA is asking us to do them. And it really comes to the point that Ira made, because many people who have excess fat in the liver are going to do just fine, like Ira said. They may have other issues, heart attacks, et cetera. But the issue is, how do you pick out the people who are going to have progressive liver disease and are progressed to cirrhosis and potentially need for liver transplantation? When FDA made their guidance in 2018, the only test that really had enough data to support predicting who was going to get worse with progressive liver disease was the degree of fibrosis in the liver as determined by a liver biopsy. So they set up a guidance in 2018 that we all have to operate under that says you have to pick out the people with a liver biopsy who are going to have stage 2 or higher fibrosis, which is kind of moderate to severe fibrosis. And those are the people who are going to target for treatment. Those are the people who are going to get drug development. Well, it's, as Jeff, I'm sure will be glad to elaborate, this is not a welcome procedure from a patient perspective. It's invasive, you can have bleeding, you can even die from it. So fortunately, there's been a ton of research that's happened since then that have looked at non-invasive tests. And I actually advocate we don't use the term MASH anymore, Alan, because what I'm talking about is at-risk MASLD. So who among the people who have excess fat in the liver are at risk for progressive liver disease? And you don't need a liver biopsy anymore to do that. There's other non-invasive tests, serum tests, imaging tests that can select people for increased risk of progressive liver disease. And I believe, and I imagine many people on this panel will agree, that that's where we should be going in terms of future drug development, selecting people with non-invasive tests.
Søren, you have your hand up.
Yeah. So I think first I would like to say, I think in this discussion it's very important that we distinguish between F2 or 3 MASH and also the more severe cirrhotic MASH patients, because the regulatory framework and the challenges are very different from these two populations. In terms of the use of biopsy as an endpoint in F2 or 3, we view that this is not really aligned with the current clinical practice where non-invasive tests are being used largely for diagnosis and stratification of the patients. And I think there's a lot of data from clinical trials. There's a lot of data from observational studies that suggest that actually this non-invasive test is sufficient in the F2 or 3 population. If we look at the F4 population, of course, the challenge is a little bit different here, because here we're looking at clinical outcomes for approval, which of course is a hassle because the event rate is very low, and so you need very long trials in order to prove it. But again, here there's a lot of ongoing trials that could validate potential surrogate biomarkers. So the hope is that we will see some validation of those biomarkers, and that will have the HbA1c of liver cirrhosis.
Stephen?
Yeah, I mean, I would just endorse what my colleagues have said, but the other points I'd make is that even though the liver biopsy might be a regulatory sort of gold standard, we also know it's a bit hit and miss, and that you can hit the liver on more than one occasion and get completely different results. So all of this staging that you're talking about doesn't necessarily hold true. Now, yes, you can have multiple sections and what have you, but ultimately it's painful. Patients don't want it. It's unreliable in terms of its accuracy. So and also the point that you've made, Søren, is that it's not actually used in clinical practice anywhere near what it's used in clinical trials. So it's a very false situation. But that's where we are. But as I think we're going to get to, the non-invasive tests are now coming into their own. And we have non-invasive tests where we can look at both metabolism, fibrogenesis, and also fibrosis. And that is helping a lot. And that's what clinicians use. That's what, and I'm not a hepatologist either, but that's what hepatologists are using, along with imaging, non-invasive imaging. So it's a strange situation that we find ourselves in.
Yep. Tony?
Yeah, I'll just add from a clinical point of view, our hepatology section very rarely uses liver biopsy, right? And clinically, it's just not there. So for all of the reasons, so it is fascinating from a regulatory perspective, pre and post inclusion, and finding patients to do that, putting patients at risk when potentially better tests available. And I think too, the other point is there's probably a panoply of diseases involved as well in determining who's going to progress, who's not. That's the real work to be done. Yep. And then who needs therapy. But clinically, I completely agree that that's just not happening.
So we have a little bit of an anachronism here in terms of time. The clinical field is more advanced than the regulatory requirements for drug development at this point in time. But Jeff, you've been leading an initiative at the Global Liver Institute to address this topic specifically. You want to just comment about that?
Yeah, thank you. When we talk about diagnostics for MASLD and MASH and fatty liver disease, to me it kind of breaks into two different categories, if you will. One is the regulatory standard, which we've just spoken about. It's biopsy. It requires biopsy. It's not reliable. All the faults of it are the reason why it's not used in the clinical situation. I do think there probably is a place for biopsy down the road in certain severe cases. We'll never be away from it, but practically it's just not what is used for patients these days in a clinical situation. When we think of diagnostics, on the other hand, and I mentioned this earlier, we're talking about risk stratification to truly appreciate the disease state. And I appreciate that a lot of the burden here that we're talking about on this panel is kind of identifying progression into the fibrotic state, as Ira said. I think that's absolutely true. But with non-invasives, if we're able to establish, I would say, surrogate endpoints, which I think is where the emphasis needs to be as we move through creating a non-invasive standard, both for clinical in the guidelines that are coming out, as well as something that could hopefully trickle up to the FDA. That's really about what gets people into the care pathway. And so it could be as simple early stages as a FIB4 test, which is done on the enzymes that everybody knows anyway. It's just basically a little bit of math. And I have patients that are part of our network and our system that were diagnosed with, you got a little bit of fat on your liver. We're going to focus instead on these other things. And then 9 years down the road, they're told, no, your liver is cirrhotic and you're going to need a transplant within 9 months, or you're looking at a very desperate situation. And so what are the diagnostics that we can use for managing and screening at that point? And there are several, that we can do. If they have an inflated FIB4, then perhaps we move them into an ultrasound, which isn't perfect by any means. But then if we continue to see this, at that point we've got them into a care pathway. We have them into a diagnostic pathway, and that's where they're going to be best served.
So the key is identifying the person who needs to be followed.
Yes.
Right?
Yeah. I just want to re-emphasize something that Mark said, because it's really important, and it's the concept of at-risk MASH. To give you an idea, I just wrote a grant, a clinically related grant, and I talked about MASH in the grant, and I had a hepatologist read it, and they said, you can't use the word MASH. That's a pathological diagnosis. So we went back and we changed everything to at-risk MASH, and it is incorporating everything that you've heard on this panel, where without a pathological diagnosis, we can do the best we can to decide who we want to go on for. So at-risk MASH is a very, very important term.
Or at-risk MASLD.
Or at-risk MASLD, perhaps even better.
If I may put a last statement onto that. For patients, these are clinical designations that could be meaningful, but especially in early diagnosis, are not. And so what the patient will hear is at risk, and then they'll begin to kind of fade away, whether it's at-risk MASLD, at-risk MASH, at-risk fatty liver disease. What they're going to hear at that point is at risk, what do I need to do? And that's where we start moving them through the pathway.
Okay, thank you. Thanks for that clarification. Again, the patient voice here is particularly important for disease where, again, symptoms aren't necessarily present when the person should be identified. But back to my three pharmaceutical development colleagues here, what then are the major impediments for getting a drug approved for the treatment of MASH with fibrosis? Mark, you want to jump again?
Sure. So the liver biopsy is the standard right now, right? So it's a major impediment. I mean, if you're a clinical investigator at a site, you have to talk to a patient about getting their liver stuck with a pretty big needle. So the first barrier is the patient says, no, thank you. I don't want that done. And if they say, okay, I'll have it done once, but then you have to say, no, actually, you're going to have at least two because we have to see if the treatment gets better. And then that's a barrier, right? Then the variability that Steve brought up. There's a placebo response. Why is there a placebo response? Well, because if you stick the liver here and you stick the liver there, you might get different fibrosis stages. And the first time it might be worse than the second time. So actually, it looks like it got better, but really, there hasn't been a treatment effect at all. It's just you stuck the liver into different places. So you have the variability, you have the patient barrier, and then you have the pathologists who look on the microscope and have to grade the disease, and they have scoring systems for grading these diseases. Well, one of the key features is something called ballooning. It's what happens when liver cells die. They kind of blow up like a balloon. And there was a paper published a couple of years ago. They got the 10 of the best liver pathologists in the world together and had them blindly rate a bunch of slides and say, circle the balloon cells. And they couldn't agree. They just couldn't agree. So we have to then have consensus among pathologists. So we have to have 2 or 3 pathologists, and they have to either agree or have some kind of voting system. It's a mess.
Søren,
add something, Søren?
Stephen?
I fully agree with that. And then if you gave them 2 different biopsies from the same patient, they would be even worse off because they would probably look different, those 2 biopsies. There would be some heterogeneity. And that's also one of the problems with the biopsies. But I think also, so we have a number of programs that is targeting the F4 population. And here, of course, it's the big problem is that you have to wait for these clinical outcomes. And that creates very long and very expensive trials. And that's one big problem. And of course, we have patients here waiting for drugs because there are no approved drugs in F4 MASH. So
under those circumstances, is the buck worth the bang? I mean, does it make sense at this point in time, knowing what we know, to pursue targeted therapy for F4?
I think it does because this is where people really get sick and where the mortality rate goes high, in particular when you go into the decompensated F4. I mean, the need there is huge. And I think whatever we do over the next 20 years, probably the rate of generation of new F4 patients, we probably won't affect it because we talk about a huge number of patients here. I think if 2 or 3, it's an estimated maybe 100 million people in the world with that. So I think it's definitely worth it. That's where the unmet need is the largest.
Stephen?
Yeah, so I was going to just say that the unmet need is significant. And it's not as though this information is just going to appear in 5 or 10 years' time, that the FDA are going to change their minds. It's going to take companies like ours and academics and people working in the field to innovate and push the boundaries so that we can help inform the FDA and the regulators so they can make those decisions. So yes, it's worth it because there's a massive unmet need out there for patients, but also we have to help the regulators understand as we develop our understanding of the disease.
We call that dialogue.
Yeah.
Tony?
So, the other point I was going to make is that we're talking about these biopsies and what we need for regulatory approval. We're currently in the middle of a phase 3 study where we're looking to get approval for GLP-1 for F2, F3. And although to get an initial approval, a conditional accelerated approval, we need to show improvements in terms of liver biopsy, as Søren is saying, ultimately it's clinical outcomes. So you move into a study that's 240 weeks or 5 or 6 years. Halfway through, it's about getting the biopsy result, but then you've still got to continue and show improved clinical outcomes. So you're asking about what's the hurdles. It's a big task to take something.
What the clinical outcomes are for the audience?
Sorry?
Want to mention the clinical outcomes
Well,
for the audience?
I mean, the clinical outcomes are that you will develop cirrhosis, that you will develop cardiovascular disease, you may develop hepatocellular carcinoma. These are the risk factors. These are the things that would happen. You may develop something called portal hypertension and all the consequences of portal hypertension. But this is what we have to show as part of a development program to enable to get a full approval of a medicine that we can then use to treat these.
And if I could just raise one other point for the audience, and these guys know this better than I do, adding to these challenges is a placebo effect in MASH trials. So if you just take people and put them in a trial and put them on the non-treatment arm, a substantial percentage of them will look like they're getting better. And that makes these trials even more challenging. Would you agree with that?
Yeah, very much so. I mean, the thing that we've noticed in the trials that we've been doing is the massive placebo response rate.
Yeah.
Right.
More than we've seen in any
Right.
other clinical development program.
Right.
Tony?
Yeah, my one push to the companies is to do far more diverse clinical trials. Most of the patients are white so far in these trials, and we've got to do a far better job of including more diverse patient populations, which makes your job tougher, but is really, I think
you'll
really
see that in
important.
phase 3 more than in phase 2.
But still, if you look at the different trial and others like that, it's not.
Well, and the FDA now has new
Right.
diversity
Right.
requirements.
Yeah.
So let's move on now, because until recently, there were no approved therapies, not surprising given the development challenges. Now there is one, I've already mentioned it. And I'm going to turn to Tony first, because surprisingly, perhaps surprisingly for all of you, this is an agonist for the thyroid hormone beta receptor. And you say, wait a minute, we're talking about fat in the liver and fibrosis. So what's with that, Tony?
Yeah, so we all make thyroid hormones, right? Many of you in the room might be on a L-thyroxine replacement. And I kind of tell the medical students they're the elixirs of life, T4 and T3. They circulate and they really regulate our temperature and our energy levels in every cell in the body. And they work through a keyhole called a receptor that comes in a couple of different flavors, an alpha and a beta flavor. And thyroid hormones circulate and bind to these receptors in different tissues, such that the beta receptor is more predominant in the liver, the alpha receptor more prominent in the heart or in the skeleton. So when there are clinical syndromes of hyperthyroidism when we make too many thyroid hormones, and that causes very fast heart rate, weight loss, can cause a high temperature and a variety of other things. And obviously, the other thing, though, that what happens with lots of thyroid hormone is weight loss and a decrease in fat. So if you could maximize that effect and minimize the off-target effects, well, wouldn't that be great? So for over 50 years, people have been trying to design agonists to the thyroid hormone receptor, which were identified not in 1986, and the first person to do that, Bjorn Wenstrom, just died last week, a couple weeks ago. But so we know the receptors, we know their molecular structure, and then through a number of different companies, initially from John Baxter and Tom Scanlon, who identified the first thyroid hormone beta agonist, to Carabio, and now to Madrigal, been able to design specific agonists for a receptor that lives in a particular tissue. So Resdifra, resmetirom, is specific for the liver. It gets taken up by specific transporters into the liver, and it activates the beta receptor, which lives in the liver, and doesn't circulate in other tissues in the body. So what this does is it allows the beneficial effects of thyroid hormone on turning up the furnace to burn fat in the liver and get rid of some of the precursor toxins that lead to hepatocyte dysfunction and ultimately to MASLD, et cetera, and all the consequences thereof. Thyroid hormone is also antifibrotic, so it actually diminishes fibrosis. And so then again, by targeting the liver, you're able to turn back the disease. Remember, in the clinical trials, the phase 3 approval trials, less than 30% of patients improved on the therapy in terms of their endpoints. So it doesn't treat everyone, but it's the first approved drug based on this unique principle of trying to target the beneficial effects of thyroid hormone to a single tissue. There are other companies around as well, targeting thyroid hormone to the brain and to other areas as well in drug development to treat disease. So also the other beneficial effect of thyroid hormone in the liver is lowering LDL cholesterol. And again, so in patients on this drug, their lipid levels fell. They didn't lose weight, but their lipid levels fell, and they improved their liver scores in terms of steatohepatitis and fibrosis. There are some side effects, but relatively safe. So it was a big, and it was about a 12-year odyssey for Madrigal, from identification, 2013, first paper, phase 1, 2014, and then to approval in 2023.
Ira, you've been very interested in pathways that lead to liver inflammation, fibrosis, mediated by fat to some degree, if not more than some degree. Do you have any insights in terms of, first, molecules that are later in stage development in terms of targeting this disease, and also other pathways that we haven't much heard about from a clinical perspective that may be of interest as we move forward?
Sure. So I think just to lay the groundwork, you've heard about the importance of fibrosis in MASH, right? So that's what really drives disease. And by the way, as Alan noted, the major reason people die of MASH is because they get heart disease. And that is also correlated with fibrosis. So fibrosis is clearly the target, and that's consistent with everything that you've heard. And so a logical question from all you guys would be, well, what causes the fibrosis? And I mean, it's obviously going to be very complicated, but it's remarkably focused in the sense that there is one dominant cell type in the liver that makes the collagen, that makes the fibrosis. And that is a funny sounding cell called a hepatic stellate cell. And it's a neuro-derived cell that's in our liver, that is there in a normal liver, hopefully everybody here, that stores vitamin A, believe it or not. But when we start to get into these metabolic derangements, the cell undergoes a transformation, and we call that activation. And those so-called activated hepatic stellate cells no longer store vitamin A, but what they do is make collagen. So it would be logical to think about targeting hepatic stellate cells. Now, there are several ways you can do that. One is to think about drugs that directly act on hepatic stellate cells, or to think about other processes that eventually activate hepatic stellate cells. And a perfect example, Tony, is resmetirom, right? And so if you look at it, it says it's antifibrotic, and you could teach us about this, but one of the ideas is the lipotoxicity that it seems to suppress represents lowering molecules that can activate hepatic stellate cells. So, Alan, to answer your question, some of the drugs that are out there now by orthogonal ways do affect these collagen or these fibrosis-producing cells, but I think an exciting area of research is to come up with molecules that directly cause the hepatic stellate cells to become deactivated. And there's a lot of work in that field, and I think the thinking is combination therapy, right? So if we have a lot of great drugs that really focus on the metabolic arm of MASH, and if we can combine those with drugs that work on the fibrotic mechanisms, particularly hepatic stellate cells, I think that could be a very potent combination. And there are a lot of exciting areas that are going on: cell therapy, RNA therapy, liver-directed siRNA therapy, but those are the basic principles.
Any particular molecules other than the incretins, which we're going to get to in a moment?
Yes, so there are lots of interesting molecules. I'll mention one that happens to be in a clinical trial, and that's galectin-3. What the heck is galectin-3? It's a protein that can bind to a site on these cells, these hepatic stellate cells, and cause them to be activated. So there is a inhibitor of galectin-3 in a clinical trial. That's one of 20. I mean, we could spend a long time talking about it, but that's 1 in 20 that is out there that's being looked at. There are many others and many exciting. And what's exciting about this is cell crosstalk, right? So the liver has lots of different cells. You know about hepatocytes. We just heard about hepatic stellate cells that are macrophages, and these cells crosstalk to each other. And some of those cells are easy to target. So hepatocytes are easy to target. Macrophages are easy to target. So you can target those cells to stop making things that activate hepatic stellate cells. And all of that is going on in either preclinical or early clinical research.
Søren, quickly on this, because I then want to turn to the...
So I think, again, I'll return to my first point, is that we also have to distinguish here between the F2, F3, and the F4 population, because the metabolic modulator seems to be fairly effective when we look at the F2, F3 population. But as we get to the later stages of the disease, to the F4, I agree with you, then suddenly the activated stellate cells and the inflammation and the senescent cells and the portal hypertension, suddenly there are some other mode of actions or some other pathomechanisms that are driving the disease forward. So I think we would probably need different tools to different segments of patients.
And that's where the combination therapy, as Ira just stated, may be particularly important.
I'll just mention one other thing, just because it's relevant to that. You might say, well, what about trying to get rid of the collagen that's already there, getting rid of the fibrosis that's already there? And so that represents another area of research. There are enzymes that can break down collagen. That's a complicated area, but there is research going there as well.
Control is important on that one.
Right. So you could imagine you had a metabolic drug, a drug that tries to turn off these hepatic stellate cells, and a third drug that tries to break down the collagen that's already there, right? So those are exciting ideas for combination therapy.
But speaking of exciting ideas, I mean, we have 3 companies here, Novo, Lilly, and BI, that are each pursuing different incretin hormones, GLP-1, GLP-1 GIP, and a triple agonist, and GLP-1 glucagon in that order. You want to comment about the incretins and why they're so appealing to treat NASH, including NASH with stage 2 and 3 fibrosis?
So, well, I can go first. Is that okay?
I went first last
Oh,
time.
you went first. Okay, I go first then. I mean, the reason at least that we are very enthusiastic about this class of drugs is that they're quite effective. I mean, what we are seeing in the clinical trials with regard to reducing steatosis, with regard to reducing fibrosis, and also treating MASH. At least in the F2 or 3 population, of course, we can see now when we look at the published data, and I'm not so surprised about it, is that it seems like the efficacy becomes more limited when you go into the F4 population. And I would expect that also because you can see that the steatosis does go down when you go from F2 to F3. But the other reason why we're also very enthusiastic around this is that we have to understand also that you have a lot of interconnected diseases, the cardiovascular, renal, metabolic diseases, very interconnected here, where having one increases the risk of getting another disease and amplifies another disease. And what these drugs also do is that they give a very robust weight loss. And of course, obesity is a key driver, so this is very interesting. They have anti-diabetic effects. That's also very interesting because having type 2 diabetes increases the risk of getting MASH. And then, of course, we also start to see hints of very strong kidney effects and so on. So I think that we should more consider a holistic treatment of these multimorbid CRM patients. And therefore, drugs that are doing multiple things become very interesting. And that's why I think these drugs are very interesting. And of course, we also believe, for instance, that the glucagon component is very interesting because glucagon has a very profound effect on the liver, increases beta oxidation. That means lipid oxidation, further contributing to the reduced steatosis. And of course, you have a lot of glucagon receptors also in the kidneys. So that could potentially also help kidney disease. So multiple reasons why this type of drugs are super interesting. But Steve, I know that your company has been the first ones here.
Yeah, I mean, I think you can work it out for yourself that if you look at incretin-based therapies and you think, well, first of all, they were designed to lower blood glucose. They work by stimulating insulin secretion. We also see some significant weight loss. So you've got 2 mechanisms there by treating diabetes and treating weight that it makes a lot of sense that it's going to have an effect on the liver as well. But if you then look at the individual effects of those incretin hormones, and as I say, through insulin secretion, you look at insulin signaling, all of that can have a beneficial effect in terms of lipid metabolism, in terms of then how you can reduce liver fat. Have to remember that GLP-1 receptor agonists have been shown to have, as Søren was saying, so many effects on so many different organs and so many effects on things that are circulating in the blood, including, for example, CRP. So we know that GLP-1 also has an anti-inflammatory effect. Now, if we believe that that metabolic derangement leads to inflammation leading to fibrosis, well, then it makes a lot of sense that GLP-1 receptor agonists and incretin-based therapies will have an effect on that as well. We're all pursuing maybe different combinations of different incretin-based peptides, also including amylin. We'll see in the next, the next 5 years or so, who's backed the right horse. But going back to, I think, a previous comment, I don't think it's going to be one horse. There's going to be 2 or 3. And I think, these triple agonists, if you like, they'll have might have the greatest effect in terms of glycemia and weight, so you think, well, they might have the greatest effect on the liver. But let's see, because there are different combinations, and it's not just the effect on glucose and weight that's going to affect the outcomes in terms of metabolic liver disease, whatever we want to call that, metabolic liver disease.
Mark?
Yes, I agree completely with Søren and Steve's comments, but I would just, for the audience's sake, give you this picture. You said obesity is a driver, right? And I do believe that the increase in fat in the liver is a driver for this triggering of inflammation, activation of hepatic stellate cells. So if you just address what Ira was talking about without addressing the underlying problem of obesity and excess fat, it would be like trying to stop a tsunami with a wall. And some of the water is still going to get over the wall, right? You have to address the underlying problem of excess fat in the liver to treat this disease effectively. Now, does that mean everybody's going to be treated effectively? No. And that was the point that Alan was making about some patients may need combination therapy. The point that Søren was making about people with more advanced disease may need combination therapy. But there may be other reasons, right, for why people may need combination therapy. There may be underlying genetic factors that predispose them to increased inflammation in their liver. And these things are just beginning to get teased out. And I think there will be precision medicine approaches coming where we can say that not only do we have to address the fat, but we have to also address this underlying problem that's driving inflammation in the liver.
That raises two interesting questions. One is it now time to think, for the first time, of preventing the development of NAFTL, NASH, and progression by intervening earlier in terms of the glycemic dysregulation that is known as diabetes and obesity? And is that one arm of this two-pronged approach where the other arm is really focused on treating individuals with more advanced disease, where that may not be sufficient, where there may only be, as with Rezdiffra, a 30% response rate? Do you have thoughts on, Tony?
I can mention, I think what's going to be interesting, we know in the United States with the coverage now with GLP-1 agonists, substantial part of the population, that if you look at the incidence in screening for the disease, which I expect to fall significantly, especially when we get towards the analogs and the pills and everything else like that. That's going to make the medicine far more available and to many more people. So that's going to be very important, I think, in terms of who we have left to treat. Do we just have the massive inflammation and fibrosis like patients who are driving disease through an alternate mechanism or a more susceptible mechanism? And those may be the ones that will then need combination therapy. One other point, there was a preclinical study with a glucagon T3 chimera, thyroid hormone coupled to glucagon, which was very effective. Probably the most effective drug preclinically in terms of treating fibrosis. So I think combination therapy is I agree from a clinical point of view.
So I think we're on the cusp of seeing something really interesting where the public health elements of this in terms of getting appropriate treatment for those who are obese and who have diabetes early enough that we can actually begin to curb the rising epidemic of this liver disease. Now, we have a long way to go because the number of individuals, despite the wild sales figures for the incretins, is still very small in terms of the population that would potentially benefit from them. Stephen, I cut you off there?
No, I was just going to say that I think you gave a statistic at the beginning that 250 million people have this condition, and by 2030 it's expected to double. And how do we prevent this happening? And I think to your point, if we treat people with diabetes, people living with diabetes, people living with obesity, with the right therapies or with the therapies that are likely to prevent this, and I know we haven't shown that yet, but if you use things like GLP-1 receptor agonists, other incretin-based therapies, where you are hitting the root cause, hopefully you are dealing with the metabolic defect that you're talking about, Mark, that maybe we can get towards prevention, maybe we can bend the curve, but there's then going to be that other population at the end of the spectrum,
absolutely,
the other end,
yep,
where we are going to need some very specific therapies. But I'm sort of cautiously optimistic, and I know that these therapies I'm talking about in terms of GLP-1 receptor agonists are not having widespread use. They're increasing a lot at the moment, but they're not hitting all the patients that maybe we would like them to, or benefiting from all those patients are benefiting. But ultimately, this could help us with the problem that we have.
You know, it raises, I'm sorry, I was just going to say it raises a very interesting point related to what Mark said, that in a thought experiment, if you could prevent the lipotoxicity, the obesity from the very, very beginning, you could reach that end. But at what point does the MASH process become self-amplifying, right? And I think that that's a very important concept. And it certainly overlaps with the high levels, but even at the early stages, there may be a series of self-amplifying effects where you're past the lipotoxicity stage. So yes, it would be great to prevent the obesity in the first place, but to the extent that that can't be done in the hundreds of millions of people, then at what point do we need to combine that with something that gets at some of these other mechanisms?
And has already been stated, can you identify those individuals who are at particular risk for that outcome, the self-perpetuating fibrotic inflammatory process that leads to end-stage liver disease? Jeff, you had a...
Obviously, I'm not a scientist here, and so I won't pretend to reflect any of these perspectives. They sound great.
we can reflect anything.
But I want to pick up on something that Stephen said, which I think is the transformative point right now, which is there is a sense of optimism. For so long, we have been Sisyphus pushing the rock up the hill in terms of dealing with liver disease, in terms of dealing with metabolic dysfunction, in terms of dealing with obesity. But when we take a look at these factors that these gentlemen are speaking about, in terms of antifibrotics, where we have hope, where we're beginning to get therapies on the market, we have the rise of the GLP-1s. We're talking about combination therapies. I think, or frankly, when we look at the pathways, we're talking about combination diagnostics as well. When we talk about prevention, we're talking about combination interventions. And that all leads to a real sense of optimism, I think, and I don't want that to be overshadowed by the puzzles that we're trying and the challenges that we're trying to solve here right now, but that there's a real sense of optimism, I think, for the first time since I've been involved in the field, that there are answers available. And we don't want it to be like, Winston Churchill had a famous quote about during World War II about Americans said that they would absolutely do the right thing after they exhausted all the other possibilities. We see this on the horizon, and if we are intentional and in good partnership, this is achievable.
It may be soon that incretins replace fluoride in the drinking water.
I just want to add one thing. I think this needs to be a both and, Alan. I mean, treating obesity is critical, and the three companies that are on the stage also have massive efforts to try to change the way people look at obesity as a disease entity that deserves treatment. So we're all in that game, right? But it needs to be a both and. I have a friend who trained with me in endocrinology at the University of Virginia. He's a practicing endocrinologist in North Carolina. And in an advisory board setting, he said, why do I need to diagnose MASH? If I just treat obesity and diabetes, isn't that all I need to do? And I disagree with that, because for the points that Søren's made about people with advanced disease, there's a good study showing that people with diabetes, about 14% of them have stage 3 or stage 4 fibrosis. Stage 4 is cirrhosis. And those people need extra care. They need, specialty care. Otherwise, they're going to end up really sick and in urgent need of a liver transplant. So treating the obesity may not be enough for that person. So we still have to focus, like Jeff said at the very beginning, and that Ira was saying, on finding the people who need that more focused therapy at the same time that we're trying to address the underlying public health problem of obesity.
And that raises really interesting questions about, back to diagnostic tools. Because, it's pretty easy to assess increased fat in the liver. Less easy to differentiate stage 2 from stage 3 fibrosis, or even stage 3 from stage 4, although that may be a little easier using the right technology. But what are the opportunities for innovation in the diagnostic biomarkers that will provide insights? And Søren, I know you have interest.
Maybe just one comment here, because the tools we have to segment the patients are extraordinarily crude at the moment. I mean, it's simple histology. We're looking at, oh, ballooning cell, cool, we note that down. But last time I checked, there were bulk RNA-seq data from 27 F4 patients, something like that. So of course we need to understand this better. And the data that we have looked at suggests that this is a very, very heterogeneous population where you see all kinds of different pathways being dysregulated in different ways. So I think you'll probably reach a situation fairly soon where you see that there will be need for multiple drugs targeting different pathways in order to treat all these patients.
Yeah, I think you've already heard about the heterogeneity in this population just from the response rate of drugs that look like they're actually quite efficacious, but only hit a less than half of the population. But Stephen,
So I think you asked earlier about, or just before now, about new biomarkers and non-invasive tests. And I think as we all do the trials that we're doing, we're getting more and more markers. We're measuring more and more things. We're learning more and more about the disease. But also, you correct me if I'm wrong here, there's somewhere between 7 and 10 public-private partnerships all collecting data on biomarkers in Europe and in the US. And some of these are now going forward for FDA regulatory review. And I think that the field will change a lot in the next 3 to 5 years as we get a whole series of biomarkers that will help us diagnose, help us stage, help us assess response to therapy. Your sense of optimism, I have a sense of optimism around the biomarkers.
Yeah.
Alan, I'll just mention one other topic that we haven't just mentioned, and I think we should, and that is, where does genetics come in? And can that be helpful in combination with biomarkers to suggest people that may be more at risk? And one of them, which is the most potent, is a gene called PNPLA3. It's much more common. The mutation that, or the variant that is a risk for MASH is more common than most people think. And to the extent that that can be used to help stratify, I think is something that's part of the conversation.
well, I think this is where the field has to get a little more clever in terms of doing future-looking clinical trials, particularly as we face the possibility of combination therapy. Which individuals are most amenable to treatment with a particular regimen based on something, whatever that something is, that will allow whoever the sponsor is to move forward in a reasonable way. The need is clearly there. The patients are clearly there, but identifying those patients with the right biomarker in the right center to actually conduct the clinical trial may not be there yet, and I think that's where we have to go.
And then to the patient's perspective, and I'd love to know your opinion, is adherence to therapy. In our health system, 65% of people who start GLP-1 agonist therapy stop within the first 6 to 7 months, just because they stop taking it. So again, talking about multiple combination therapies, complicated therapies, difficult for patients to adhere to.
Yeah, very much so. I mean, we've never done prevention very well. For any of us that have tried to wake up early to go get a walk or a workout in, I mean, it's really been difficult. And so adherence to medical regimes without having, appropriate support is something that becomes that much more challenging for patients to do on an individual level. Patients with this disease state tend to be fairly isolated. We just flew 100 liver disease patients and caregivers into Washington for an advocacy training, and many of them had never met anybody with their disease state, had never met anybody with MASH before. And so when you get into, adhering to the medical regimen, we would advocate that having partnership with organizations like ourselves or with local organizations to be able to provide the support to check in, to be able to do that. That gets into design a little bit, I think, to be able to be helpful for that, because it's hard for all of us.
It is indeed hard for all of us.
Just one more comment on biomarkers, Alan.
Yes.
So biomarkers sound good, right? We don't want to do liver biopsies, get a blood test, get an imaging test that's non-invasive. It sounds great. But these tests aren't perfect either. And so, and there's different contexts of use for biomarkers. So some biomarkers are better at diagnosis. Some are better at prognosis. telling you how sick that person's going to be. There's biomarkers that are better for treatment response. And so that science is still evolving. And where it is right now, most experts are saying you probably need agreement between two biomarkers to be really confident what's happening with the patient. Is the patient at risk? If there's two biomarkers suggesting the patient's at risk, then they likely are at risk, right? If they have, if they get treated and two biomarkers get better, you're pretty confident that that person's getting better. But what do you do if one biomarker gets better and the other one doesn't get better? So you have a conundrum, right? So that field is still evolving, and like Steve mentioned, there's a lot of these big consortia that are working on getting these biomarkers more adequately studied and validated.
So, we've talked about the challenges of getting drugs approved for this condition because of the need for biopsy. I'm curious about what the panel feels about the regulatory acceptance of the concept of a prevention indication. Now, we're not talking about using the drug to prevent the disease, because that's being done now in diabetes and obesity. We're talking about prevention of MASH with fibrosis as an indication. Is that possible? We do such a bad job in terms of getting regulatory approval for anything for prevention. Stephen?
I think we'll have one step at a time, please, Alan.
Okay. Come on. You can't shut off the discussion that quickly.
I think we found it a big enough challenge, just trying to get a pathway where we can we can design a clinical development program so that we can get some medicines approved in what clearly is a big area of unmet need. I think moving into prevention is going to be very, very challenging, even more challenging than the current challenge that we're facing.
Well, I mean, Steve, prevention of diabetes.
Yeah,
that's,
exactly.
That's not an indication.
No, exactly.
I mean, why not? It should be, but
Right.
it's very, very hard to study to do that trial.
And here it would be even more difficult, I think.
Yeah.
Oh, absolutely.
Because
That's why I brought up
earlier
the
fibrosis
question.
stages are completely asymptomatic. So I think it would be hard.
Was that meant to be a rhetorical question, Alan?
It was a little bit of a rhetorical question, but it also I meant it to be provocative from a public health perspective, because we just do such a poor job in terms of preventing disease with pathways, with mechanisms, with lifestyle modification that we know works, and yet society hasn't.
We think
gone in that
we all agree it would work, right? We all agree
absolutely.
we can prevent it. It's just that how do you do that trial?
And we have to maybe change the economics of our healthcare system.
Yeah.
For sure.
We don't pay well for prevention.
No, we
I mean,
don't.
it's going to take us, the best part of 5 years to demonstrate clinical outcomes in people with the disease. Then to throw some prevention into that as well.
Of course, with prevention,
We'll have even more grey hair by the time we get there.
So, Jeff, last question to you before we open it up to the audience, and I'm happy for everybody else to pitch in here. How do we increase awareness of this disease enough to have an impact in terms of the outcomes for individuals who have it? What needs to be done?
There are two ways. One is increasing education and awareness is necessary but not sufficient. And so, as we've seen with other education and awareness campaigns and efforts and whatnot, it takes more than just say no or let's move in order to get people aware and acting on their disease states. In most public health campaigns that have focused on this, they have also been backed up by strong advocacy, regulatory, and legislative efforts. We take a look at what's happened with tobacco, for instance. We know that that was a multi-pronged effort that combined a lot of these aspects. There's really, from our show of hands of people that just knew where their liver was, there's a lot about basic liver health that I think we can do better education with. The patients, certainly, as a patient advocacy group, we're trying to do this one person at a time to educate them, not just to be able to, frankly, have the conversation with themselves, with their families, with their providers, with the FDA. We realize in those conversations that primary care is a huge issue right now. So many primary care providers do not screen for liver disease in this way as part of their regular stuff. That's reflected in the lack of coding in the AHRs that we need to do better improvement on. And so I would say, yes, we have to have better education, and we achieve some of that through partnership with some of our partners, like those that are on stage right now. But if we don't run that in parallel with education and advocacy around the systemic issues on this, within healthcare and within biopsy and non-invasives, then we're going to fall short of this. Those are the two prongs that we have to move forward with.
Okay. Happy to take questions from the audience if there are any. Please identify who you are if you have a question. Great.
Hi there. Great panel. Rajashi Banerjee from Oxford. I want to address your friendly endocrinologist, Mark, who said just treat the obesity and the diabetes and don't worry about the liver disease. I sympathize with him or her a little bit, and I'll explain why. Once you open the can of the liver disease, there's a lot of Latin, yeah? And Latin doesn't translate well to the patient interaction. So my plea to the panel is when you next come to talk to the FDA, please ask them to, even if it's just by half, reduce the amount of Latin by half. And that's coming from a guy who's educated in Oxford. The second thing is, if you do this thought experiment, if you had someone with obesity and hypertension, and you just treated the obesity, and they still had hypertension, there would be no doubt in my mind that I should also treat the hypertension, even though part of that may have been treated with the obesity treatment. I still think there are some latent biases against liver disease, and Jeff, you may have some thoughts about this. But the big thing for me in addressing these biases is, and maybe it's not enough people know where their livers are, but they all know where their hearts are. And liver disease is a massive modifiable risk factor for heart disease. So if you can't persuade me to take my drug for my liver because I don't know where it is, you can persuade me to take your drug to prevent me having a heart attack. And so I do think that when you do the regulatory affairs meetings with these drugs and these indications, please bring in a couple of friendly cardiologists just because they're more comfortable with the bigger trials and they're not so laser focused on just the liver outcomes and the Latin. So that was my two pleas.
You know, if I could just comment on that, I think that that's a really important point, and you've heard almost all of us talk about why the #1 cause of death in liver disease. I study heart disease. That's where I began, and then I moved over to MASH about 10 years ago. I'm now applying grants to the National Heart, Lung, and Blood Institute on the two together for exactly the reason that you said, and the AHA has come out with a statement echoing what you just said, and so it's easier for people like us to get grants funded that look at the links between liver disease and heart disease. And I think from Jeff's point of view, if that message can get out, that would be very powerful.
I'll be the defensive optimist here, again, defending my optimism, which is this nomenclature change from non-alcoholic to metabolic is still pretty fresh. I mean, we don't have it in our EHRs. We've got coding scheduled for October a year from now. And so I think the promise of that, moving away from non-alcoholic to metabolic, is now we are creating those relationships, we are creating those pathways, we are creating those connections where now the liver community is in the conversations with the heart community, with diabetes, with obesity, with other stuff. And so I think you will see that coming.
I think the concept of considering both diabetes and obesity, multi-organ disease with liver disease, with heart disease, with kidney disease, is one that really needs to be refreshed in medical schools all the way through. There was another question over here. This may be our last.
Stanley Weiss from the Rutgers, New Jersey Medical School. I'm an MD and epidemiologist, and from that perspective, I think you've had a very interesting session that may be missing some opportunities for data analysis from what you have in hand and how your companies could collaborate. And I think it involves changing your nomenclature and how you're interpreting things. You're trying to convince the FDA not to rely just on liver biopsies. You have evidence that it may not be the right gold standard. I would posit that when you're using the term placebo response, that many of those patients were misclassified. So there is a misclassification bias. Well, you can look at your endpoints that you have and do a retrospective cohort analysis to identify what other markers you have in other studies, and also identify, since you know those people haven't been responding, and then you could posit that some of those people were misclassified. So if you do that on several of your studies as your test exploratory data sets, you could then apply it to other studies which have those same tools and look at the predictive value, having already justified it. We do that all the time in epidemiology and various other cohort analyses. I think you've got the opportunity, and that then you could do it pretty efficiently and convincingly.
Great. Thank you very much. I want to thank the panelists for their contributions today. A lot of thought, a lot of good discussion. And I want to thank you, the audience, for giving us the opportunity to opine here about NASH and MASH and all things liver, diabetes, and obesity. Thank you very much.